Hi everyone. In today's session, we are going to be talking on building haplotype network, and mainly the software we are going to be using is the popArt software. All other softwares we are going to be using in this session would the link to downloading those files will be provided in the caption section. So this is the popArt software. In order to build this the network, we essentially need two files. We need the alignment file and the trace file. So let's build these files. We have our sequences here. We have 12 sequences in all. In order to add more sequences, provided you needed to add more sequences, you simply need to copy your sequence. Once the sequence is copied from the source, then you can come and import here. But for this example, we are simply going to be using 12 sequences. So once you have it, the next thing we want to do is we want to run a multiple sequence alignment. We are going to be using the molecular evolutionary genetic analysis software, more like the mega software. So we come to File, open the file, then we select the sequence, which is our file name. It will bring this option, align or analyze. We'll select align. So while we have this, our sequences have been imported into the mega platform. Then we want to, the next thing we'll do is we'll select all. We'll come to this edit option, then we'll select all. After selecting all, we'll come to this. We are going to be using a module algorithm to run the multiple sequence alignment. So select module and we'll align our samples or align our sequences. Press OK. So once our sequence has been aligned, the next thing we want to do is to export this aligned sequence. So we come to data option, then we'll export data. We'll export data as Nexus format. So we can save this data as aligned sequence. We are saving it on the desktop. Save. So once you're done with that, you can close the mega software and also the Bioedit. So this is our saved aligned sequence. The next thing we want to do is to build the haplotype data file. And to do that, we use DNA sequencing polymorphism software. This is the software. Again, the link to downloading the software will be provided in the caption. So we'll select file, then we'll select open data file. We'll come to all data files. We'll select all files. Then we'll select align sequence.nexus, more like the align sequence. Once you select that, you open the file. This is a generalized information about our sequence, largely. Then we'll close the data. Then we'll come to data, select format. The basic information about our data is our sequence. The sequence we are using as an example is a mitochondrial cytochrome oxidase subunit 1 sequence. So the sequence is the mitochondrial part and it's haploid. So select mitochondria, haploid, and OK. The next thing we might want to do is to assign genetic code. If you're sure of the source of your sequence, it fits from a Drosophila organism, yeast, flatworm, you can select that. But for this example, we'll be using the nuclear universal. Once we are done with that, then we'll define sequence set. So let's assume now we have 12 sequences like we said initially. So H1, let's assume H1 to H5 is from say another location, let's say Kenya, we can, after selecting, and we press this forward sign here, we can add a sequence. We say Kenya. Okay. The next thing we want to do is we assign for the other location. Let's assume this location is from, this sequence is from Nigeria, and let's assume from 10 to 12 is from, say, Ghana. So we have Kenya from H1 to H5, Nigeria from H6 to H9, and Ghana from H10 to H12. Once we confirm that, we update all our entry. The next thing we want to do is to generate the haplotype file. We come to this generate option, this haplotype data file. We select that. On this platform, we select included and press OK. It will ask you to give an option for you to save the haplotype data file. So we can save this as haplotype data file. Then we press again. You're saving it on the desktop. Select save. Once you are done with that, we can close the DNA sp software. So the next thing we want to do essentially is to copy the file to which we need to create our traits file. So we open the haplotype data file you just saved, then you open it with Notepad. The next thing, after opening it, you will see this the first option here will be the list of the haplotype, the number of sequences present in each of the haplotype. Then we have a more breakdown of the content of each of the haplotypes. But the one you would really need to copy essentially is the list, the one that has the list, the frequency of the haplotype, and the sequences, the name of the sequences. So we'll copy that. Once we copy that by simply pressing Ctrl C, or we'll right click and copy, we'll go to our Excel sheets, then we paste the file. So we have this file pasted. The next thing we need to do is to create a table. app_1. So far we have 7 haplotypes. We'll drag it to make sure it's 7. Then the first location is Kenya, second is Nigeria, and third is Ghana. So recall that we said Kenya has the sequence of H1 to H5. So H1 and H4 is under Kenya for each haplotype H1. So we put 2 here. H8 to H9 is under Nigeria because H6, we've already assigned that H6 to H9 is Nigeria, and Ghana is 10 to 12, so 2. H2, which is under haplotype 2, is H2, which is Kenya. H3 is Kenya. H5 is Kenya. H6 is Nigeria. H7 is Nigeria. H11 is Ghana. So the next thing we want to do is to fill up this space with zeros. So once we are done with that, we'll copy all. Then we'll open another notepad. Then we'll paste it there. Once we do this, we'll backspace. Then we'll put comma, backspace, comma. In front of the haplotype, put comma, 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 comma. Put comma, 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 comma. Put comma, 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 comma. Then in front of each of the numbers, we press tab, 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 and tab. So once you've done this, we'll save our file. We'll save our file as traits. Then we'll save it. We'll note again we are saving on the desktop. So once we are done with this, we can largely close our file. And we open the popArt. So we import our alignments, which will be the, so once you select imports, is to bring this option, then you select all files. The file you have to select is the haplotype data file. Select that. To bring this option, select OK. Then you come to this import, you import the traits. Simply select traits, clear data, clear alignment data, no. Simply press OK. So once you have this, you build your network. So largely, this is the network. You can use different algorithms to build the network. I initially used CCS. I can use some other algorithm to build it, depending on my targets. So you can also design further by using any of the design that you desire. So this haplotype distribution can also be viewed on map. By coming to view, you view it on map. As you know, this is Nigeria, this is Kenya, and this is Ghana. So you can actually use this to give a better description of your sample location as well. So you can go back to view the haplotype network. Then you can change the background to white. Change the background to white if you want to copy the image. Then you save you export the graphics. You can prefer to use any. Let's use PNG. You can save it as image. Then select save. So once you do that, your file has been saved. You can come to your desktop to view it. Taking time. And that's it. Largely, that is all about building haplotype. If you have any, feel free to leave a comment section and I'll be quite ready to give a response. Thank you.